Hello guys, and welcome to Brainstorm Biology. I'm Victor and I'll be taking you guys on cell biology. So, with reference to our last video, we'll dive in to explore some of the organelles in cells and also their functions within the cell. Come with me as we explore that. In our last class, we were able to discuss about the cell membrane. We discussed about proteins embedded in the cell membrane and also the roles of these proteins. Now, in today's class, we'll continue from where we stopped and we'll be looking at organelles such as the cytoplasm, nucleus, and other organelles of cell. So let's start by explaining what cytoplasm is. The cytoplasm is this region of cell that is occupied by fluid or that is occupied by mostly water. So the cytoplasm in terms of composition is mostly water, and this is where all other organelles are suspended. So this is where other organelles such as the endoplasmic reticulum, Golgi bodies, the mitochondria are embedded. Now, we'd like to talk about the cytoplasm in details. The cytoplasm, as I said earlier, is mostly water. So the cytoplasm in terms of composition is mostly water. And the other major chemicals that form the cytoplasm aside water is proteins. So in terms of composition, cytoplasm is water and protein. And anything that is composed of water and protein has the consistency of jello. So it is jelly-like. If you were to feel cytoplasm in your hand, it is jelly-like in consistency. Now, to bring this home, if you've ever noticed this, is actually common among those that are into cooking. You agree with me that after boiling meat, the water obtained is jelly-like in nature. And that is because meat is high in protein, and the proteins in meat begin to dissolve in water as you boil it. So that creates that jelly-like fluid or jelly-like liquid obtained after boiling a meat. So that liquid, that is that jelly-like liquid, is the same thing in terms of composition to how cytoplasm would feel if you were to feel it. So that is that about cytoplasm. It is mostly made up of water and protein. And that is that about the cytoplasm. Another thing worthy of note is inside of the cells there are proteins. There are proteins inside of the cytoplasm, and they are referred to as cytoskeleton. Now, if you can recall, from the beginning of the class, I explained what cyto means. Cyto is an anatomical terminology used to refer to cell, while skeleton, just as the name suggests, is a framework or a structure that supports. So cytoskeleton basically means a structural support for cells. Now, in addition to the cytoplasm, in this cytoplasm there are proteins that serve as skeleton of a cell. So just like we humans have skeleton that gives us form and shape in the form of our bones, at the microscopic level, there is what we refer to as cytoskeleton that gives cell its shape and form. And these cytoskeleton are categorized into three. We have the microtubules, microfilaments, and intermediate proteins. So structurally, the cytoskeleton are proteins, that is in terms of structure. Microtubules, just as the name suggests, is a tube-like protein. It is tube-like in nature. So microtubules as a protein is kind of tube-like in nature. Like a pipe, hollow in the inside. Microfilament, on the other hand, is just like a thin wire. So it's just a solid, thin protein that is like a wire. And intermediate protein is protein that is between the microtubules and the intermediate proteins. So that is that about the proteins. And one of the characteristics of some of these proteins is that most especially microtubules, they can move. So their ability to move is what gives cells its ability to move also. And the movement of these microtubules are what causes cells to move. And the movement of cells are referred to as um, either cytoplasmic streaming, amoeboid movement, or we can call it contraction. We have more to talk about regarding the microtubules as we go forward. Now, the next thing we are going to be looking at is the nucleus. Now, the nucleus basically means center. It comprises of a nuclear membrane. Let me point it out. Nuclear membrane. Nuclear membrane. Now, the structure of the nuclear membrane is similar to that of the cell membrane, which is it is composed of a bilayer of phospholipid molecules, and just like that of the cell membrane, it is similar to the nucleus or the nuclear membrane. Another thing about the nucleus is that it possesses openings or pores that allows for exchange of materials between the nucleus and the cytoplasm. So it has openings in it that allows for exchange of material. The third thing you have to know about the nucleus is that it contains what we refer to as nucleolus, which is a dark region in the nucleus where RNA is synthesized. And lastly, just like the way the cytoplasm is mostly water and protein, the nucleoplasm, which is the liquid found in the nucleus, is also water and protein, 80% water and protein. And if you were to fill the nucleoplasm, it will also feel like jello, or it will be jelly-like in consistency. Now, in addition to the nucleoplasm are DNA. So, in addition to the nucleoplasm is DNA, and we know that DNA are genetic materials that are transferred from parents to offspring. So the DNA controls all activities of the cell. Now, I want to clarify on something. There are terms we use to refer to DNA, and we have chromatid and chromosomes. Now, these terms chromatid and chromosomes are referring to the DNA, but they both mean different versions of the DNA. Now, the chromatin or the word chromatid is used to refer to DNA when it is uncoiled. So DNA as a genetic material can coiled up, which is in this form, or can be uncoiled. So when the DNA is tightly coiled, we refer to it as chromosomes, which is in this form. While if it is uncoiled, which is in the form of like a spaghetti, we refer to it as chromatid. So basically, chromatid and chromosomes are just terms used to refer to whether or not the DNA is tightly coiled up or uncoiled. So that is that about the nucleus. Now the next organelle we'll be discussing about is the endoplasmic reticulum. Now let me simply break what that means. Endo is actually referring to inside. Plasmic, inside of the cytoplasm. Reticulum simply means a network of tube. So the endoplasmic reticulum simply means a network of tubes inside of the cytoplasm. A tubular network inside of the cytoplasm. That's what endoplasmic reticulum means. Now, surprisingly, there are two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So, the rough endoplasmic reticulum, smooth endoplasmic reticulum. Now, the rough endoplasmic reticulum is considered rough because it is rough. The smooth endoplasmic reticulum is considered smooth because it is smooth. And I will explain that. You see, the rough endoplasmic reticulum is studded, or it has what we call ribosomes. So it is studded with ribosomes or has granules on it. That is why we give it the term rough. While if you notice, this endoplasmic reticulum is smooth because there is nothing like ribosomes on it. That is why it is referred to as smooth endoplasmic reticulum. Now, both endoplasmic reticulum have their functions. The smooth endoplasmic reticulum is involved in lipid metabolism. And by that, we mean it is involved in the breakdown and build-up of fat or lipids. So, lipid metabolism. That is the role or the function of this smooth endoplasmic reticulum. It's involved in the breakdown and also the buildup of fat. While the rough endoplasmic reticulum, on the other hand, is studded with ribosomes. And one of the functions of ribosomes is that they are used in synthesis of protein. They are used in gathering up, or they are used in the formation of protein. So proteins that are formed in the rough endoplasmic reticulum are then transported. That is transported inside of this rough endoplasmic reticulum. The synthesis of protein occurs in the ribosomes, and then the ribosomes transfer those proteins into the rough endoplasmic reticulum for transportation. So that is basically that about the endoplasmic reticulum. We have the rough and the smooth. While this is focused on the transportation of synthesized protein, this, which is the smooth, is responsible for lipid metabolism. That is the breaking down and build-up of lipids. Another organelle I would like us to look deeply into is the Golgi body. Golgi body. Now, the Golgi body, or some people call it Golgi complex, others refer to it as Golgi apparatus, is simply a series of pancakes. It's like a pancake that is found in the intracellular environment of the cell. Now, what the Golgi body does is that proteins that are synthesized or made in the rough ER, and also fat synthesized or made in the smooth ER, are transported into the Golgi apparatus or Golgi body for temporary storage and modification. So what that means is that proteins made are then transferred into this stack of pancake-like structure, and they are temporarily stored and modified. Now, if you can recall, we discussed about one of the proteins found in the outer, that is the cell membrane, in the outer part of the cell membrane. We discussed about glycoproteins. Now, how glycoproteins are made is that proteins that are made from this rough endoplasmic reticulum are transported into the Golgi body, and then the Golgi body modify it by joining it with a sugar molecule. So it not only temporarily stores these chemicals in the form of proteins, it also modifies it. And in modification, that is how we have, or we come to obtain, glycoprotein. So the Golgi body serves as a temporary storage of chemicals and also it serves as a modifier of chemicals. Now, the next organelle we'll be discussing about is the mitochondria. The mitochondria is said to be the powerhouse of the cell. So all the energy needed to sustain a cell is actually produced in this organelle known as the mitochondria. And one of the interesting things about mitochondria is that it is the only cell or only organelle rather that possesses its own DNA. So mitochondria has its own DNA outside of the general DNA that is found in a cell. So this is a region whereby ATP is being produced. And that is the function of the mitochondria. Again, let me just briefly explain how this process occurs. Now, organic molecules such as sugar react with oxygen to produce carbon dioxide and water plus energy. Now, this is typically explaining the reactions that occur in the mitochondria. So, this is what we call cellular respiration. Now, if you can see clearly, there are enzymes. There are enzymes within the mitochondria that help in catalyzing all these reactions to produce that energy in the form of ATP. So, when organic molecules such as glucose ignite with oxygen, it produces carbon dioxide and also water as a byproduct, and most importantly, produce energy in the form of ATP. So about 32 molecules of ATP are produced in this structure known as the mitochondria. That is why it is referred to as the powerhouse of a cell. Another important organelle we'll discuss briefly is the lysosome. Now, the lysosomes are organelles that contains digestive enzymes. The function of the lysosome in a cell is that they aid in digestion of food particles within a cell. So, lysosomes aid digestion. And one of the things about lysosome is that they contain hydrolytic enzymes. And these enzymes, when ruptured, that is when they get out of this sac-like membrane, they can lyse out the entire cell. So because they aid digestion or they contain enzymes that are capable of digesting food particles, if these enzymes are to get out of this confinement, which is this sac, they can destroy the entire cell. They can digest the entire cell. So lysosome is not just limited to digestion, but we'll see briefly some of the roles of lysosome aside digestion. One of the roles of lysosome is it is responsible for the formation of our fingers and toes. And by that I mean lysosomes are responsible during their programmed cell death, or the genetic information from the gene as programmed cell death through the use of lysosome. But then, before I explain that, I would like to talk to you about a term we use in referring to the actions of lysosome. We have apoptosis or autolysis. Now, apoptosis is a term we use to refer to when this lysosome rupture, that is, they release out their contents and digest an entire cell. So, this is the term we use to refer to that situation, and also this is similar to it, just that I would like it to be more clearer to you. Auto simply means self. Lysis means to break apart. So autolysis simply means the lysosome releasing this content, leading to the breaking apart of the cell. So why exactly is this structure very important? As I said earlier, it is responsible in the programmed cell death during the formation of fingers and toes. Now, one of the functions of the lysosome is that during embryonic development, at about 5 weeks of development, the embryo's fingers, or the embryo's toes and fingers, do not really form. In other words, it is like a paddle-like arrangement of skins or cells. So there is no segregation of fingers during that development. Now, ideally, there are cells that were programmed. By programmed, I mean the instruction of the gene. They were programmed to commit suicide. Committing suicide simply means they were programmed to autolysis. In other words, some of the cells are genetically programmed for this lysosome to rupture and digest the entire cells. So the vertical column of cells that were programmed, when this lysosome rupture and release their content, they digest all the vertical cells that are arranged here, and that's what brings about a separation. And that's what brings about a separation. Likewise, there are cells also, vertical column of cells that are genetically programmed for this lysosome also to rupture and digest the cell. That's what brings about another separation. And that's how we go from a paddle-like fingers or paddle-like hands to fingers and toes. So that is one of the important role of this tiny little structure known as lysosome. They are not only involved in the digestion of food, but they are also involved in the formation of fingers and toes. Another important function of the lysosome is that they cause what we call atrophy. Atrophy of the uterus during menopause. Now, what this means is that first I would like to define the term atrophy. Atrophy simply means shrinking. So shrinking of the uterus during menopause is also caused by these organelles. Now during menopause, the woman's ovaries stop working, and by stop working, they stop secreting the female hormone, which is estrogen, and also they stop ovulating, which is they stop ovulating their egg. That is, the egg process in a woman finally comes to an end. And while that occurs, the uterus or the cells lining the uterus atrophy, and the atrophy, as I said earlier, is simply to shrink. So the atrophy of the cells, that is, as the lady approaches menopause, which is a stage where she no longer gets pregnant, is as a result of this lysosome. So the lysosome rupture and then releasing their content in the cells, digesting the cells, and that causes the cell to shrink. The shrinking of the cell signifies that the womb or the uterus cannot hold a child, or it cannot mother a child. So these are also the role, and the actions of this lysosome is what led or leads to development of osteoporosis. That is the weakening of the bone in women. So you can see that lysosome is involved in the formation of our fingers and toes, and not just limited to that, it is involved in the shrinking of the uterus at menopause in women. Lastly, another role of lysosome is that it's atrophied, or it digests a cell that is infected with virus. One of the things about viral infection is that it is very, very difficult to treat. In fact, there are no vaccines, or there are no medication for viral infection. So when a virus gets into the cell, The only way that our cell can destroy that virus is by allowing this lysosome to rupture, to release out its content and digest the entire cell. And as it digests the entire cell, it also takes down the virus along. So you can see that the lysosome, as tiny as it is responsible in the formation of our fingers and toes. It is responsible in menopause it's responsible in the atrophy of the uterus during menopause, and most importantly, it is responsible in the digestion of cells that are infected with virus. Now, the next other organelles I would like to discuss about are cilia, flagella, and centrioles. So I will discuss them together because explaining them actually covers everything about them. So first of all, what is flagella? Now this is a typical human cell, sperm cell to be precise, and you can see there is this rope or whip-like structure, and that is what we call flagella. Now cilia, this is a paramecium cell, a single cell organism, and you can see this hair-like projection. It is what we call the cilia. And the centrioles are responsible for cellular division. That is, they are the ones that aid in the separation of chromosomes during cellular division. Now, before starting, if you can recall, while I was talking about the cytoskeleton, I said we'll have more to discuss about on microtubules. So, microtubules as proteins are the fundamental units of movement. So, this flagella cell, this cilia, and also the centrioles are all composed of microtubules. So, let's try and see what exactly is inside of the ciliated cells, and also what comprise the centriole. So if we were to make a section or to cut this and see what is inside, it will look something like this. This is what it will look like. All these tube-like structures are the microtubules. And this kind of arrangement is what we refer to as 9+2 arrangement. So it is these microtubules that aid the movement of this flagella cell. So it is what gives the flagella cell ability to propel and move towards or in a fluid environment. So you can see that microtubules are the fundamental units of movement. Similarly, if we were to make a section to see what is inside of this cell, this cilia, you also find out that it comprises of this kind of arrangement. So the cilia is basically a short version of the flagella. So it's still made up of these 9, which is 9 pairs, 1, 2, 3, 4, 5, 6, 7, 8, 9, plus 2 in the center, 9+2 arrangements. These are the fundamental units that enable this organism, which is paramecium, to move. And there is another one, which is the centriole, having 9+0 arrangements. Now, in the case of the centrioles, there is nothing at the middle. So it is just arrangement of microtubules round, that is having a 9+0 arrangement. And that is the fundamental and basic unit of movement in a cell. Now, speaking on the cilia, the way that we develop as human beings is as, first of all, the sperm has to fertilize the egg in the fallopian tube. And one thing about the egg, or one of the features of the egg, is that it is immotile. It means it cannot move on its own. So there has to be something to move it. And the moment fertilization occurs, the egg has to leave the fallopian tube and then implant in the endometrium of the uterus. So in the inner lining of the fallopian tube, there are cilia, which is cells like this, if I'm to, let me just give a picture. So if fertilization was to occur here, there are ciliated cells that lines up the inner membrane of the fallopian tube, and it is these cells which is having the 9+2 arrangement, these ciliated cells having 9+2 arrangement, that aid in the movement of the egg down to where it implants in the endometrium. So you can see that microtubules as proteins are the fundamental unit of movement both in ciliated cells, in the human cells such as the sperm, and also they are the cells that aid in the pushing of the egg from the fallopian tube down to the endometrium where it implants. So that brings us to the discussion on the flagella, cilia, and centrioles. With this, we've come to the end of today's section. Don't forget to like, subscribe, and share. See you in my next video.